Welcome everybody to our weekly research seminars. Today, it's my pleasure to introduce our speaker, Dr. Babak Razani from Washington University. Dr. Razani has received 2 bachelor's degrees, one in electrical engineering and computer science, and the other in molecular and cellular biology from the University of California at Berkeley. He then went on to earn MD and PhD degrees at Albert Einstein College of Medicine. And after completing internal medicine residency and a cardiovascular fellowship at Washington University School of Medicine, he has stayed on as faculty, and he was recently promoted to the rank of associate professor. Dr. Razani has conducted seminal work in the field of atherosclerosis and metabolism. He was the first to establish that the autophagy lysosomal system becomes defective in atherosclerosis, and has described the pathogenic relevance of cytotoxic protein aggregates that accumulate as a consequence of this. His most recent work on stimulating the transcription factor TFEB as a means of driving the autophagy lysosome biogenesis shows the potential power of enhancing this pathway in ameliorating cardiovascular and cardiometabolic diseases. Dr. Razani's efforts have been recognized by the scientific community. He's received several prestigious early career investigator awards at national forums, including the Irvin Page Award at ATVB, the David Williams Award at Cochran, and the Springer Award at NAVBO. So it's our pleasure to have you today, Dr. Razani, and we look forward to your talk on the dietary protein and cardiovascular risk. So the floor is yours.
Oh, super. Well, thank you, Murray. I really appreciate that introduction. And again, Murray and I go way back, multiple, visits to ATVB over the years, right? And a lot of overlap in our works, which would be, which has been really great. You've contributed, quite significantly to this area as well. So what I wanted to talk to you about today. And too bad, by the way, I can't be there in person. That would have been cooler to see, everybody in the same room. But this is sort of one of the topics that we've been working on that's kind of taken a life of its own. It dovetails with the autophagy lysosome system in some respects. I'll talk to you about that. So yeah, I want to tell you about dietary protein and atherosclerosis and cardiovascular risk. Really for this audience, this is unnecessary, but just suffice it to say that atherosclerosis, we all recognize as a lipid-related disease. That's not a shocker. It's sort of a dogma. And everything from obesity, diabetes, and other cardiometabolic risk factors impinge on this lipid idea, right? The driving of the plaque is predominantly lipid related. So we don't have to look far other than a lot of epidemiological studies, of course, and intervention ones, with the statins in particular, that if you drive LDL levels down, you will drive cardiovascular risk down as well. Now, one thing to note is that this line is not a 45-degree angle, so it's not a one-to-one relationship. And in fact, there's a point of diminishing returns where you have to really drive down LDL below to be able to get a sort of a marginal effect, right? So that's important to note, but nevertheless, it is, every time they've looked at it, it's sort of a linear association. This is a figure from all the genetic type of data, including the PCSK9 mutations that were discovered in the past decade that, of course, now have led to the PCSK9 inhibitors. Same type of situations. You bring LDL levels down, you will drop cardiovascular risk to the point that you could argue that if we have normal lipids, and by normal, I should say, not in the 80s, 90s milligram per deciliter, really in the 30s, 40s from birth, that could potentially prevent all of cardiovascular disease. So you could make an argument that this type of intervention or keeping those levels down from birth is really the way to solve the issue. But that's not where we're at. There's a lot of people with this disease. And what you're going to act on at the moment is people with plaques that have already been generated in their 30s and 40s that are progressing. And so LDL is not the entire story. Again, as I said, there's a point of diminishing returns where if you drop it too low, you're not really getting much more bang for your buck. So we got to look elsewhere. There's a lot of avenues that, of course, many people are looking at. But we got interested in a nutrient angle just because then we could dovetail it with the LDL hypothesis. And we were interested, what about other nutrients in our diet, and how do they impact atherosclerosis? And so the question came into dietary protein. And it's been sort of interesting with dietary protein because a lot of people use it as for its metabolic benefits that we'll get into a bit. But the issue is, what does it do to the cardiovasculature? So to look at this, let's just drop back and just talk about what are the general recommendations for our dietary protein intake. This is now from U.S. Department of Agriculture kind of guidelines. I'm sure in Canada you have very similar types of recommendations. And so a lot of people talk about dietary reference intake, and most people agree that 0.8 grams of protein per kilogram of body weight is really the level we should be eating. So that equates to about 56 grams a day for men, 46 grams per day for women on average. And again, the USDA guidelines actually are largely similar to what the Mediterranean diet is. So I went down and looked at a bunch of diets. Here's USDA recommendation. Here's Mediterranean. Weight Watchers, Atkins, South Beach, Zone, Paleo, Optavia, Dukan, Wolverine, Keto, Ornish, so and then there's a bunch more as well, right? Currently, there's a keto craze, of course, going on. But for the longest time, many people they talk about weight loss diets or metabolically beneficial diets. They're talking about elevating dietary protein. So here's the USDA, here's Mediterranean, right? This is where the recommendations currently lie, which is you're talking about a 15 to 18% protein intake, 29%, 30%, about a third fat, and in the 50s of carbohydrate, right? Now, you notice with these other diets, what has happened is they dial up the protein, right? 26% is what Weight Watchers is, all the way to get to some crazy stuff like the Wolverine, where I guess you become a Wolverine, right? You're eating proteins, 50%, or whatnot. Really just massive amounts of protein intake, right? I wanted to compare this to the keto diets, where you notice the protein is actually fairly low. It's just lard you're eating, right? Look at this. This is crazy. And I don't know who's taking a keto diet, but I'd be very concerned about this diet, right? With the particular fat intake. A lot of people swear by it for weight loss, of course. We don't know its cardiovascular benefits or risks. And then here's Ornish diet on the other side, where it's really a high carb diet, all these low. So this is very instructive to kind of take a look at this. But suffice it to say, lots of people swear by this. I'm sure lots of people in this room or in this Zoom meeting maybe are, part of this equation, right? You're taking this kind of stuff. And then, then we go to when we were teenagers, and maybe nowadays, a lot of you guys do more workouts too. I don't as much, right? Which is bad, but we'd go lift some weights, if you remember, in the teen years, and then you're taking 2 scoops of protein. So right after your workout. So look at what's happening. I mean, those protein smoothies that you guys drink, is giving you 40 grams of protein, right? That's essentially, if you will, doubling what you're taking on a daily basis, what's recommended, right? So we have generally a lot of protein in our diets. I could give a whole talk about what the protein craze is in this world. And it's quite fascinating what people eat, and they overeat, particularly to take the protein, right? So there's a lot of health benefits that's been purported from protein intake. Obviously, it's a favored diet, notwithstanding the keto craze. favorite diet in many weight loss programs. And people think that because of the weight loss, there's a secondary improvement in insulin resistance. And that's why people like this as a metabolic fixer. Now, it's a very controversial topic, but there's been reports of cardiovascular risk with dietary protein as well. And, you all would agree with this, that, of course, the red meat issue has been out there for a long time, right? People swear that fish and chicken proteins and other things, and of course, vegetable type proteins are much better than red meat proteins. The controversy here is that red meat in particular are laced with a lot of fat and lipids. So when you take that, you're really accentuating your diet in a lot of nutrients, not just protein. So the controversy lies, is it just red meat is a problem, or is protein a problem, right? Well, there's been several studies on this. There's maybe 10, 15 decent ones. Here's a really good one from a group in Sweden, looking at 43,000 Swedish women, prospective study. And as you know, doing a prospective randomized study on protein is very difficult, right? Because now you're trying to control people's diets, nearly impossible. So these are largely questionnaire type of studies, but it's still prospective. They follow and they see what people are eating, and then try to come up with some conclusions on what the nutrients are doing. So you will notice that here is a low carb, high protein score that they came up with. As you increase this score, that means you're taking more protein in your diet. You will notice that your incidence rate of cardiovascular disease it elevates, no doubt about it. Huge number. And this type of observation has been replicated in several other type of prospective studies. So the signal is there. And it's potentially a concerning signal, especially because of the dietary craze that we're facing. Then, when we got interested in this, we saw this kind of epidemiological data. We said, well, let's see what's happening in animals and really animal models, I guess you could say. And it's interesting that if you actually just start Googling this stuff, you'll notice that there's entire books written on this subject. Here's one, Dietary and Proteins and Atherosclerosis from Debris. Lots of good data in here about rabbits, mice, pigeons, you name it. This guy assessed that, and there seems to be a clear association between protein intake and plaque progression in all types of animal models. The question is, where's the mechanism? What is the mechanism? And how well are these studies designed? And that's where the issue comes in. I think you will notice that the literature is replete with a lot of observations, but little mechanistic insight. And so that's where we are. And we decided to get into the game simply because, ourselves, Mireille, lots of other investigators in your esteemed institution, you guys look at mechanisms of atherosclerosis, right? So that's what we were interested in the context of dietary protein. So what we did is we designed a specific diet here. You'll notice this is our standard tech-led diet that a lot of people use. You'll notice that its protein content is about 15%. Interestingly enough, the Western diet that tech-led sells is giving you protein that a USDA would recommend, right? What's different, obviously, is that its fat content in kcal percent, is 42%. That's really what's driving the atherosclerosis. We all wouldn't debate that. And so in this diet, it's really a carb fat diet, relatively low protein. So what we decided to do is change the equation, keep the Western diet part of the diet the same. So fat content is exactly the same. Obviously, we don't want to change that because you muck around with the fat and you're going to get changes in atherosclerosis that are completely dependent on lipids. We tripled the protein. So now we're getting into a Dukan Wolverine type of situation. And then we're at 11% on the carb. So it's a low carb, high protein type of diet, same fat. So what you'll notice is same thing with mice. As in humans, the mice don't gain that much weight. So it's very nice. They resist weight gain. The standard Western diet guys gain a good amount of weight. And these are, by the way, I should note, these are ApoE animals, lipoprotein E knockouts, which are standard, one of the standard atherosclerosis type models. The other being LDLR that a lot of people use. So GTTs, glucose tolerance test, you'll notice again, very beneficial effects. So taking this by all measures is metabolically beneficial. Mice eat it well, and they don't gain too much weight. But what's interesting is what's down here, which is really the plaque assessment. You'll notice despite all those nice metabolic benefits, you end up getting quite a bit of atherosclerosis. Now, the lesion size itself is about, 25%, 30% higher. It's really what's instructive is on this side, which is somewhat on the macrophage contents is up to, but it's really the tunnel staining, the necrotic core formation. These are the ones that are up about twofold. So a lot of features of plaque complexity is driven by this excess protein in this diet. So this was quite surprising because you have really a disconnect, right? From great metabolic benefits as assessed on the diabetes and obesity spectrum, but quite an increase in atherosclerosis. So we were wondering what exactly is happening mechanistically. As I said, a lot of us are interested in mechanisms, are not really just observations. And we would want to leverage these mechanisms for potential future therapies, which I'll get to in the end of the talk. So one thing that we, I guess it's not too much of a shocker, is that we would anticipate that when you have dietary protein, you're going to have increased amino acids. And when you have increased amino acids, you're probably going to drive mTOR. And so we had a hypothesis. Is mTOR really a signaling pathway that gets stimulated in these organisms? And is this the one that's the pathogenic mediator of this dietary protein? So first thing we looked at, and I'm not going to show you a lot of this data. This is published about a year and a half ago in Nature Metab. So I'll show about 3 slides of this and then move on to some of the translational work that we've done. But suffice it to say that if you look at blood monocytes, you look at splenic macrophages, you look at plaque macrophages, and we looked at other tissues as well, by the way, liver, fat, etc. You end up getting quite an acute stimulation of mTOR activation. So this is using S6 as one of the downstream effectors of mTORC1, which is the pathway that's activated by amino acids. And you'll notice there's quite an elevation of phospho-S6, a downstream target of mTOR, and it comes down. Similar in splenic macrophages, similar in plaque macrophages. And if you look at aortic roots, so these are atherosclerotic mice on a high protein diet, you'll notice that when they eat, in general, you get a lot of co-localization between CD68 and phospho-S6 again. So really alluding to the fact that on an acute basis, if you're eating dietary protein, or on a chronic basis, if you're on a protein for several months, you get this activation of the mTOR signaling pathway. And there's a lot of other figures in that paper that you can take a look at as well. So then the question was mTOR really the driver of all this phenotyping? So what we did is we went to classical genetics and knocked out Raptor, which you all know is one of the major components of the mTORC1 complex, as opposed to the mTORC2 complex, which is driven by a Rictor-dependent process. So a lot of people, when they study mTOR signaling, they'll either knock out Raptor to get rid of mTORC1, or they knock out Rictor to get rid of mTORC2. And by the way, we have a paper that we're about to submit on mTORC2 phenotyping, which is quite interesting as well, which we can reserve for another talk on that subject. But here's the Raptor knockout. This is using Lysem-Cree floxed and then Raptor floxed mice to get rid of a myeloid specific, in a myeloid specific manner. And you'll notice that the Raptor knockouts are basically knocked out for Raptor and completely oblivious to mTORC1 signaling, right? So when you try to stimulate you starve with from amino acids and you add leucine or other types of amino acids, you'll notice that there's really no signaling happening, or very little signaling. Here is another surrogate of signaling, which is the co-localization of mTOR with lysosomes. This is actually all the was all the rave in the past couple of decades, really driven by David Sabatini, who said that mTOR signals at the lysosome. And this is clearly the case, that when you starve a cell with amino acids and give it amino acids back, you get quite a bit of co-localization between mTOR. That's really the signaling hub. And that's actually a line of research that we're very interested in as well, is using lysosome as a nutrient sensing type of organelle, which is beyond the scope of this discussion particularly. But suffice it to say that mTORC1, we have a capacity to completely get rid of that. So I'm going to summarize a lot of work in this, just with this one slide, and to say that the paradigm using these raptor mice that we found was the following: that high protein diets lead to elevations of circulating amino acids, which activate mTOR signaling in a lot of tissues, but particularly macrophages of the atherosclerotic plaque. It drives atherosclerosis, and it in particular drives plaque complexity. So is this macrophage raptor dependent? The answer ends up being yes. This is really a key figure of this whole paper, is really the linchpin figure. And you'll notice that here's dietary protein driving atherosclerosis. Here is raptor knockout mice, macrophage raptor knockout mice, completely abrogate this high protein diet effect, right? So it really nails the hypothesis, right? That this atherosclerosis is driven by macrophage mTOR signaling. And all these plaque complexities are completely nullified in the raptor knockout mice. And then I will summarize again, lots of work on this paper with one slide. Mechanistically, downstream of this, of course, our favorite is autophagy. In a lot of it is my lab, but also it doesn't just have to be the favorite. It is actually a favorite signaling, downstream signaling pathway for mTORC1. We all know that it's exquisitely sensitive. Autophagy is exquisitely sensitive to mTOR signaling. When mTOR is high, autophagy is completely suppressed. And a lot of the work that our lab, Mireille's lab, other labs have participated in, have demonstrated, is that autophagy is critical to dampen atherosclerosis. And we showed that when you drive mTOR signaling through amino acids, that it ends up being that autophagy gets suppressed. And actually, in particular, mitophagy gets suppressed. And this is very interesting because now you get a buildup of really dysfunctional mitochondria, which drive a mitochondrial apoptosis picture. And I think this is what is the stimulator of the plaque complexity. So a lot of that plaque apoptosis is happening because of a dysfunction in mitophagy and an accumulation of ROS generating and caspase generating mitochondria there. So that's the story in a nutshell in mice. Very mechanistic. And again, you can take a look at that and gauge for yourself how much detail we put into this to really drive that mechanism. And you could argue it was the first paper that tried to nail what is happening on a signaling level in atherosclerotic plaques. And with that, the lay press, when we published that, jumped on the idea that dietary protein increases heart attack. So a lot of titles here and whatnot is that, hey, this dietary protein is bad for you because we showed that it increases heart attack, whereas we really never ever showed that. And obviously it was just a mouse study, but it really begot the concept that, well, what is happening in humans? We had that epidemiological data that can be criticized in a lot of levels. Of course, it's prospective, but yet it is questionnaire based. There's no mechanistic human studies around. And so we want to say that, hey, if we're going to be people that work on dietary nutrients of all sorts, including dietary protein, that we want to be translational biologists in this respect. So what's happening in the mice is it occurring to some level that we can gauge in the humans. Now, of course, with the caveat that you can't do a whole lot of things in humans to the level that you can do in mice, obviously. You can't get tissues as readily and whatnot. But you can do certain things that, that are very tractable. And this is what I wanted to discuss with you for the rest of this talk. And so one is to say that we can obviously, with human MCSF, we can take monocytes and make human monocyte-derived macrophages. That's very easy. And from this, we can actually evaluate mTOR signaling with or without amino acids and whatnot. And it turns out that human monocyte-derived macrophages are probably the best or one of the best, if not the best, cell with mTOR activation. If you take any other cells, these guys are so ready to be stimulated by amino acids to activate mTOR. You can see that already here. You starve with amino acids, you put amino acids back, you put leucine back, you put other arginine, any of that kind of stuff that are classic mTOR activators, and you get this huge response with mTOR activation. You get a decline in autophagy, and you get elevations in apoptosis when co-incubated with things that kill macrophages, like things that uncouple, the respiratory chain in mitochondria, oxidized LDL, cholesterol crystals, you name it. Amino acids in general are very bad in this synergistic way. So we know that this is a very robust system in vitro. Now, in vivo, in humans, again, us being a little bit hamstrung with getting samples, I mean, obviously, you're not going to go grab someone's carotid artery and look at macrophage signaling. You're not going to go start biopsying different aortas and whatnot, right? So that's where we're hamstrung. But what we can do is look at monocytes and circulating monocytes. Very tractable system. Lots of people evaluate this as essentially a surrogate for what could be happening in macrophages and the atherosclerotic plaques. It's not a perfect science, obviously. These are monocytes, circulating monocytes. They're not tissue macrophages. But what we do is we're sort of proud of this because my lab is, mostly early translational and even, sort of animal physiology. But this was our first clinical trial. I mean, they call it a clinical trial. It's sort of a really soft version of a clinical trial. It's really a clinical study. But the United States, at least, they try to tattoo you for anything you do on human research. So it's dubbed a clinical trial. But we just wanted to intervene with regard to nutrient status and give high protein versus a low protein setting and look at monocytes. So here is our Joe Schmo participant drinking a smoothie, high protein versus low protein. So what we decided to do initially is to try to go big. So we took a big smoothie, really a slug of protein, and compared it to a very low type of, so 50% kcal versus 10% kcal protein. 50%, by the way, kcal is about what you would take if you, after a workout, if you're doing a bodybuilder would put a couple of scoops, again, of protein in their smoothie, and that's what they get. So we very nicely were able to take, the Buffy coat from this human blood, and then look at, in particular, CD14 positive monocytes, CD16 negative. The CD14 positive is really what is accepted as a proatherogenic type of monocyte, the ones that get recruited in to the atherosclerotic plaque. And then we showed, using a variety of techniques, that we can look at mTOR signaling by FACS, by Western blot, functional assays, autophagy evaluation, you name it. So we've been evaluating this and testing all our, abilities on these monocytes to be able to create a robust framework to evaluate this exact question. So again, clinical study 1 was 50% versus 10%. So again, big slug of protein versus a very small slug. And we made a crossover trial. So when we recruited subjects, these are largely normal, healthy controls. And we would fast them initially. And then this is a crossover trial in that they come in and get either the 10% protein or the 50%. And then a few weeks later, they get the 50% so that we can compare within the same individual. And it gives you a lot more strength, statistical strength, as well as the fact that the variability is dropped as well, since you have this internal control. So not a lot of exclusion criteria, but just we didn't want people with active heart disease, and a lot of metabolic diseases, so that in case there's activation of mTOR1 in those respects, that we don't necessarily get confounded by that. So here is a summary slide of a lot of stuff we sort of evaluated and things that I'm not showing here as well. But again, this is the CD14 monocyte population. And you'll notice, very interesting right off the bat, is that in humans, just like in mice, if you give someone a 50% protein smoothie, look at the amount of mTOR activation. Here's phospho-S6 level, just quite a bit, over a 1 to 3 hour time scale, right? Maybe this is obvious, but it's actually cool that you can demonstrate this, that if you and I and anybody else goes and drinks a protein smoothie, that you're getting quite a significant activation of your mTOR1 in your circulating monocytes. And of course, we say that this is a surrogate for what's happening in tissues as well. And this is by Western, by FACS. And here is the thing I alluded to before, that again, when mTOR co-localizes with LAMP2 or LAMP1, these are the lysosome markers. That's a marker for activation. And we see that as well. So there's no debate that a 50% protein is going to be able to do this. Now, a big criticism of this study is that it's super physiologic. 50% is used by bodybuilders and smoothies and taking smoothies. And if you actually look at this figure, this is a very instructive figure. This is our protein intake. This is typically data from a bunch of studies in the Western world. And so this is what we're taking in Canada, United States, Europe, etc. RDI is the recommended daily intake, as I told you, 0.8 grams per kilogram body weight per day, right? This is the level, the bell curve of what we are actually taking in Western society. So it's very interesting. Recommended daily intake is here, but notice almost all of us are taking more than this, right? I sort of alluded to this at the beginning about the public health issue here. Lots of people believe in dietary protein. They believe they're going to become, a bigger muscle mass with this. They're going to lose weight, etc. So almost, I would imagine, everybody in this Zoom meeting is taking a bit more than they should be taking. Now, there are more than others. A lot of us take a smidge more, maybe 30, 40% more. But some of us quite taking, much more than that, right? And it tails off. Now, the 50% protein smoothie that I just alluded to is way, way out here, right? These are the Wolverine guys. So the large portion of the population is not here. So we wanted to really test a real world model. And so this is our, what I would call, let me see if I can remove this. Anyways, I would call a more real world clinical study number 2. And what we did here is we ended up doing a crossover again, but we changed the protein content into 16 versus 26%, right? And actually, these numbers really should be 15, 22%. This was a previous thing, but so the data I'm going to show is actually 15, 22%, but the labels are all 16, 26. But suffice it to say, it's more real world, right? So 16, 15% is in that level where the recommended daily intake is. This is the USDA, this is the Mediterranean diet kind of level, right? And the 22, 26%, this area is just a smidge higher. It is a level actually that correlates to about this. So I would say about 30% of the population are taking this level of diet. And the other thing that we did that was cool is we no longer gave smoothies here. We gave mixed meals. So this is an actual human being taking a meal that's fortified with a bit more protein isolates than this one is. Again, crossover, same type of exclusion criteria. And what you notice, and this is a much more nuanced evaluation, but turns out that, again, your mTOR gets activated, and a lot of other stuff get deactivated. Here is autophagy as a marker is getting deactivated. So a similar concept as the 10-50%, but just on a lower, more nuanced level in the mixed meal 16-26, 15-22 type of paradigm. So at least we kind of had a gauge on where these thresholds are. And of course, we can't do ad infinitum type of proteins unless you guys give me several $million. We try to narrow it down to 2 studies where we can have sort of a more of a handle on things. And we figured out that in the 15% range or 10% range, right, from both those studies, mTOR does not get activated. A bit higher, 22, 26, 50%, obviously, much more activated. So now what we wanted to do is look at kinetics of amino acids. So here's a bunch of mass spec data of circulating amino acids. And you'll notice, and we divided these by hydrophobic, polar, electrically charged, but really, I think in this paper that we're submitting, we have it as essential, non-essential. That's a little bit more, accepted by the community. But suffice it to say, a lot of amino acids go up and come down. This is not a shocker, but we showed it in humans as well. Now, the key point is some amino acids go up a lot more than others. And you'll notice here, I'll really allude to these. Look at these branch chain amino acids. Now, I will grant this is a whey type of protein that we're using in these. But you'll notice that leucine, isoleucine, valine in particular has, but a lot of them go up. Methionine, tyrosine, phenylalanine, tryptophan, these kind of things. So if you look at this, this is a replotting of this type of data in 10 versus 50% protein smoothies. You'll notice that the excursion of amino acids in the circulation of these patients, of these subjects, is quite significant for everything, but some go way up more than others. So we chose sort of the top 8 here of level of excursion here, okay? And again, as I said, this is heavily weighted towards branch chain amino acids and some of the other ones that you might not think about. We did exactly the same thing with the protein meals, the ones that were more nuanced and more real world. And we created a Venn diagram, and I won't go through all that data, but suffice it to say that we ended up choosing sort of the top 6, 7 amino acids that were equal in both. And it turns out that leucine, isoleucine, valine, threonine, methionine, lysine, and arginine actually is another one. And we decided to take those in 2 dose versions. One, we said, okay, let's take the same dose, 2 millimolar, which is a standard amino acid dose that's used in cell culture to activate mTOR1 signaling. And we take that same dose from each of these amino acids and we put them on human monocyte-derived macrophages and look at mTOR. So here's a very instructive finding. Maybe this is surprising to some of you, is that the only one that really signals, these guys signal a little bit. You'll notice they kind of go up. But leucine is the big guy in town, right? This guy drives mTORC1 much more significantly than any of these other amino acids. And remember, all these amino acids were specifically selected for ones that have significant excursion during protein intake. So these go up. And yet, leucine is really hands down the most activated. Now you could say, well, this is equal standard cell culture dosing. So we took these same amino acids, and now we actually gave the peak concentration of the amino acid that we actually measured by mass spec in these dietary protein studies. And it turns out that, again, leucine is the #1 activator of mTORC1, even using physiological observed doses of these amino acids. And what's interesting, by the way, and I don't show the doses here, is that isoleucine and valine and some of these other guys actually had higher excursion than leucine. So this is something like 400, I think, micromolar, and these are something like 600, 800. And yet leucine is the one that particularly activates mTOR1. So it turns out that I think you can collapse a lot of this data into saying that mTORC1 is a leucine dependent process. It's not these other amino acids. And all those, by the way, amino acids have been implicated in mTORC1 activation. But physiologically, it really looks like it's leucine that's doing this. And so we took a lot of this data from the mass spec and from the 16, 26%, 10, 50%. And we looked at the peak concentration. So at 16% protein meal and 10%, we get about 130 micromolar, 26%, 22%, we get about 190, 200. And then the 50%, we get about 400, 500. So we look at these circulating levels, and we actually put them back, the leucine, back on human monocyte-derived macrophages to see if we see any kind of an effect, threshold effect, any kind of additive effect, et cetera. So you'll notice that actually it turns out that even though mTOR gets activated, and I don't know why I lost my pointer. There we go. Oops. All right, there we go. Even at lower doses, when you get activation of mTOR, in particular, there is a jump in activation around this 100 to 300 kind of range, right around where this 22, 26% protein meal is. And if you look at mTOR with LAMP2 co-localization, autophagy inhibition, mitophagy inhibition, this is COX-4 as a mitochondrial marker with LC3, activation of reactive oxygen species, activation of apoptosis, there seems to be a jump right at this level of 100 to 300 micromolar. So notice the activation of LAMP2 with mTOR co-localization really jumps at this level. Your autophagy jumps down at this level. Again, this 100 to 300. Mitophagy, activation of reactive oxygen species, apoptosis. This is, so somewhere in this 200 micromolar range is where you start getting these damaging effects. And this is all borne out from actual circulating physiological doses of the amino acids, and then looking in human monocyte-derived macrophages for mechanistic insight. So we decided then to take this kind of knowledge and this potential threshold effect and go back into the mouse to try to evaluate some level of atherosclerosis here. And we actually did very similar things in humans on the mice. So we would gavage them with protein smoothies in some sense to look at activation. And you'll notice this is the data I kind of showed to you before, where if you gavage a mouse, you get mTOR activation very acutely. And then what we wanted to do is then put this concept into a dietary form to be able to look at atherosclerotic progression. And it turns out that obviously, if you have, so for example, let's go back to what I had alluded to before, which at the very beginning of the talk, we had a 15% kcal protein versus a 46% kcal protein, right? This is a tripling of protein content. However, what I wanted to tell you is that even though you triple the protein contents for mice, they're eating less, right? Because it's a satiety effect that the dietary protein is having. So their actual protein intake is about twofold. Using this kind of concept, we ended up designing 7%, 15%, 21%, 46% kcal proteins, and kept testing these in order to be able to come up with a nice range of dietary protein intake and try to superimpose it on this human scale and see if we can dial up the protein in a very quantitative manner and see where this threshold potentially lies with regard to protein being okay and protein being damaging. And, you know, again, I think this slide, we can debate over it for a long time on how best to evaluate this, but we looked at protein intake and quantified that and tried to superimpose it on the human curve and came out with 3 diets eventually from this analysis, 7%, 21%, and 46%, that would give us quite a nice spectrum of protein intake in mice that would correlate with what's seen in humans. And we ended up making specific diets with equal fat contents, but the dialed up protein, and gave it an ApoE mice, just like we explained before, to see what happens with atherosclerosis. And then something very interesting comes out. It turns out that only when you double protein intake, you get a lot of atherosclerosis. At low protein and moderately elevated protein intakes, nothing. And it really jumps at this level. And again, this is about twofold of protein intake in mice, which correlates to about 25, 30% of the human population, what we're eating, which is about twofold. And you'll notice that macrophage, contents, necrotic core, etc., is elevated really in particular with this. Now, interesting signal is apoptosis degrees were elevated in even this 21%, but it didn't manifest over a 2-month diet with plaque complexity as gauged by necrotic core. So it looks like it's almost intermediary trying to fall into this, part of the threshold where it becomes damaging. So from this data, and again, I rushed through some of this stuff for the interest of time, but we come up to this kind of model, which is if we dial up protein and we dial up plasma leucine levels, we end up reaching a level at which point mTORC1 starts to really rise. And this is that threshold response where not a lot happens low and a lot happens high. And this is that sigmoid type curve that I mentioned to you with regard to a lot of damaging effects of the amino acids on the macrophage. So from this data, we can clearly see that there's an area of protein intake, which is about 1.4, 1.6 gram per kilogram per day, that we get increased atherosclerosis, we get monocyte activation, all that kind of stuff. And there's an area right around this, below 1.2, where low risk, there's not a whole lot going on. This is an area that we don't know what's happening. This would require much more nuanced evaluation where you dial up protein every, 2 to 5% to see what's going on. And again, would require a lot of bucks to be able to evaluate this. But from this data, you can get an idea that clearly there's bad areas and there's good areas that you could potentially use as recommendations in public health policy. And I think the more detailed evaluation of this would be what would drive the public health recommendations in the future, particularly with mTORC1 as a surrogate marker here. So now I won't go through all this data. There's actually another paper that we have, which is comparing plant proteins versus animal proteins. I'm sure a lot of you are thinking of this, that is there a difference between these types of protein? And the answer is yes, there is. In fact, plant proteins have a lot of different activation of mTOR compared to animal. And it's a huge, interesting discussion to get into that I won't have time to on why that is. But this, I wanted to raise the concept of precision nutrition. And this is that it's not that all protein is going to be bad. It's the levels of the protein. And at what point do you consider that you're not getting, for example, muscle building, too much muscle building, but yet you don't want enough mTOR activation? So, let's say, I'll give you a practical example in the clinic. So let's say you have a 70-year-old patient that just had a heart attack, significant heart attack. They're in the ICU. They're not eating very well. The nutritionist comes by and gives them a Ensure, Boost, all these protein types of smoothies for them to be able to build muscle so they don't have muscle cachexia, atrophy during the hospitalization. But the answer is, the question is this a good strategy? Is this having an effect on their heart attack? Is this having an effect on their monocytes? And is this really having a beneficial effect on their muscle building? What are the thresholds with regard to this? So this is my concept of precision nutrition. I think we should be at a level with dietary protein where we can tell you exactly what levels build muscle, and then at what points you're not building muscle anymore, but rather you're having downsides to mTOR activation, cardiovascular risk, etc. So last pieces of the talk here is what are the therapeutic implications? So obviously, if you bought into this story, is that if you modulate then amino acid mTOR signaling, there could be some avenues of benefits we can get on the cardiovascular front. Lots of you are aware of rapamycin. There's a lot of papers showing that rapamycin, which is a classic mTOR inhibitor, are anti-atherogenic. And the big problem with, with rapamycin is that there's a lot of side effects. Some people think it also affects mTORC2 signaling. It actually causes insulin resistance, partly because of that, etc. So a systemic mTOR inhibition might not be the best thing. Although here, again, I will show you, this is our own data, and this has been published in multiple fold. When you give rapamycin to mice, a lot of good things happen, right? But there are some downsides to it. So how do we make a targeted type of version? So what we've been working on for rapamycin and mTOR inhibition is we're actually trying to create a nanoparticle that's targeting macrophages. And this is using CCR2 binding peptide, ECL1I that we call it. And this has been shown on several studies, particularly one of our collaborators at WashU, that it targets macrophages. And we're trying to now impregnate this nanoparticle with rapamycin to be able to target it more to macrophages. And then the double cool version of this is we actually have nanoparticle versions of this with copper and gold so that we can look at PET imaging and try to inject it in mice and even potentially in humans and seeing where it targets, et cetera. But suffice it to say that some of the data here is very nice. This is actually in vitro using different types of nanoparticles. You'll notice that if we take a nanoparticle that's not targeting to CCR2 and one that is, we get a lot more uptake in macrophages. That's actually in vitro, and there's some types of evaluation we're doing in vivo on this front too. And here is on the, let me see if I can, well, that's too bad. I'm not sure if we can actually start this, but anyways, this was supposed to show you some live imaging where we actually give this rapamycin impregnated nanoparticles, and we end up showing that you can stimulate autophagy and you can block a lot of the leucine effects on autophagy. So it was demonstrating that we can maintain a very robust degradative process in macrophages with these nanoparticles. So, and the last slide, and this is actually another paper that we're writing up. I don't have time at all to talk to you about this, but this is a very cool concept, is that, well, we have the rapamycins of the world, and they're very generic mTOR inhibitors. The question is, can we really target mTORC1, and can we target amino acid mTORC1 signal? And I will have to say that mTORC1 inhibition and the desire to do this very specifically is something that a lot of companies are after. There's at least 10 or 15 out there that are trying to make selective inhibitors of this. So the way we're trying to get to this is, what if we target amino acid mTOR signaling? And our approach is really trying to inhibit amino acid uptake into macrophages. There turns out to be a lot of amino acid transporters. They fall under the family of the SLC family, if you have a solute carrier family. Lots of different types that are specific to leucine and other things. And this is where we're at. And this paper that hopefully in a future talk I can tell you about is all about how beneficial this is, not only in respect to dietary protein, but really just mTORC1 inhibition in atherosclerosis. So I wanted to close with that. This is kind of the lab as it stands now at WashU. Lots of contributors to this project and others, but in you know, we need to really focus on Zhang here. He's been driving this project, and then the you know, the Nature Metabolism paper is his. The one that's being in revision right now is his, and then the animal plant one is his as well. So so he's really driving that aspect of the lab. And of course, a lot of collaborators, in particular, Bettina Mindorfer, who's helped us with a lot of these clinical studies. I mean, this was not our forte. This was definitely her wheelhouse. And so she's helped us with that and the funding there. So thank you all. And I finished a little bit later than I wanted to, but, you know, maybe we can still take some questions.
Yes, absolutely. Thank you, Dr. Razani. This was amazing. Love hearing about all your latest work. So I'll just jump into the questions from, and then I'll save the ones for the people that I know that will be at the roundtable for then. So a question from Katie Rayner. She says, excellent talk, Babak. Really, really interesting and clinically relevant findings. I'm curious if you looked at other cells in the circulation in your subjects with high protein diets like neutrophils or T cells to get a full picture of the inflammatory state of high protein diets.
That's super, Katie. Thanks for that. Yeah, so when we take the Buffy code, we start sub-selecting the CD14 positives and not any of the T cells, neutrophils, that kind of stuff, right? And, you know, in a way, we shortchanged ourselves. You're right, because if we took that, then we could potentially compare all the different types of circulating immune cells. We don't have a lot of data to suggest that there is damaging effects on T cells, B cells, or whatnot. But I think those are great areas, right? So you could easily say that those are all going to be activated. I would suggest probably the case is true. From a lot of our data, when we looked at, for example, smooth muscle cells, I didn't show this, fibroblasts, macrophages, I mentioned this on one of the slides. Macrophages just love this leucine. They activate mTORC like there's no tomorrow, you know, compared to these other cells. And so it turns out it probably has a lot to do with these amino acid transporters. They really robustly activate those, but I don't know where they lie on T cells, B cells, neutrophils, et cetera. It's a super question, and I think that would be the next type of projects there too.
Okay, thank you. The next question from Ali Amadi. Thanks for the nice presentation. Would active versus sedentary lifestyle possibly change the association of high protein intake with atherosclerotic plaque formation? Does active lifestyle have any effect on mTORC signaling?
Yeah, that's a super question. I have no idea. mTOR is anabolic, right? And obviously it shuts down catabolic type of responses. Exercise in general is associated with catabolism, but also exercise is associated with stress. And so you're going to have a combination of catabolic responses and adrenaline driving the process. And I would imagine you would not be in a state of anabolism during that. I don't know what would happen to mTOR signaling, but it's a fantastic question. I think what could be kind of cool is exercise, long-term exercise, of course, it has its own effects on lipids, et cetera. I will allude to this. This is actually another paper we have. Not sure how big we're going to make it, but there is one paper from Roberto Zoncu. If you follow him, he used to be a postdoc for David Sabatini. He now has a big lab in Berkeley, UC Berkeley. So he showed that cholesterol activates mTORC1, and he discovered a solute carrier called SLC38A9 that mediates this process. So we know that cholesterol is very activating of mTOR. The combination of amino acids and cholesterol is something we're working on. And so I think you should take a look at that as far as a synergistic effect of nutrients on mTOR. And so exercise is different, but it really eggs on that same question: is what things would synergize with amino acids to drive the process. I just wanted to allude to that cholesterol concept because this is a paper we're working on that respect.
Wonderful, thank you. So in the interest of time, I will close this now. Thank you everybody for joining in. Thank you, Dr. Razani, for joining us. And if anybody else is interested in joining the roundtables with Dr. Razani immediately now, just email me or Kelsey and we can send you the Zoom link for that, and you can feel free to join in for further discussion. Thank you.
Hey, thanks a lot for everybody for showing up.